Welcome to IB Biology Video 8, looking at Unit 1.6, Cell Division. Okay, by looking at cell division, we're going to start off looking at the process of mitosis. Now, mitosis is a process which allows cells to arise from pre-existing cells. Now, we've already said that this is one of our sections of our cell theory, that all cells arise from pre-existing cells, and it is mitosis that is part of the process that allows this to happen. During mitosis, the division of the nucleus into two genetically identical daughter cells occurs. So basically, this process enables the cell to clone itself. In order for this to happen, all the DNA in the nucleus must first be replicated, and this is done during the interphase. The interphase is not actually part of mitosis, it's a phase that occurs before mitosis kicks in. Each chromosome is converted from a single DNA molecule into two identical DNA molecules, which we call chromatids. These chromatids then become separated and end up in different daughter cells. Mitosis is broken up into 4 stages, and mitosis is only one of the components of the cell cycle. We'll look at that shortly. So mitosis creates 2 clones of the parent cell. Okay, so here we've got the cell cycle, alrighty, and we can see that we start off in this interphase, okay? Then we have mitosis occurring, and then we have cytokinesis, which is the final part of the cell cycle. So the sequence of events that occur between cell divisions, okay, that is the cell cycle. The key events that occur during the cell cycle can be divided into our 3 main categories. As I just said, we've got the interphase, we've got mitosis, and then we have cytokinesis. Okay, so it's really, really important to remember that mitosis only includes the prophase, metaphase, anaphase, and telophase. Okay, the interphase and cytokinesis are not part of mitosis. They need to occur for the cell cycle to occur, but they're not part of the process of mitosis. Can you remember or come up with a mnemonic to remember the order of the phases? So a mnemonic that will help you to remember interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. Somehow you need to remember that order. Okay, so here we've got another illustration of mitosis, and we're saying that the cell cycle starts here. We work through the interphase, okay, and then we hit mitosis here at the prophase, metaphase, anaphase, telophase, and then we have cytokinesis occurring. Again, another image now of the cell cycle, showing the interphase taking up the majority of the time, followed by mitosis and the 4 phases of mitosis, and then cytokinesis. Alrighty, let's start looking at each of these phases and what we've got going on now. So starting with the interphase, and we said this is a very active phase and there's many processes occurring within the cell. DNA is replicating in the nucleus, we've got protein synthesis occurring in the cytoplasm, and mitochondria and chloroplasts are increasing in number because this cell is getting ready to divide. So the interphase consists of 3 phases. We've got the G1 phase, the S phase where the DNA replicates, and the G2 phase. From the interphase, we're now moving into mitosis. So our first phase of mitosis being the prophase. Here, our DNA undergoes supercoiling. So from just being a spaghetti of DNA as such in the nucleolus, the DNA now supercoils, and it coils up around what we call these histone proteins. Now, as I said, DNA naturally sits as this messy string-like substance in the nucleus. You can't see it under an electron microscope until it's supercoiled into chromatids. DNA must be arranged neatly for cell division to occur correctly, so it winds itself up to form compact, organized structures, those chromatids. Okay, so our DNA has supercoiled. The nucleolus has broken down. See, that's broken down there. Microtubules grow from structures that we call MTOC. Okay, these are microtubule organizing centers, and they form spindles that link across the poles of the cells. At the end of the prophase, the nuclear membrane then breaks down.
DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. Next, the nucleosomes are packaged into a thread. The end result is a fiber known as chromatin. This fiber is then looped and coiled yet again. Leading finally to the familiar shapes known as chromosomes, which can be seen in the nucleus of dividing cells. Chromosomes are not always present. They form around the time cells divide when the 2 copies of the cell's DNA need to be separated.
Okay, so still now looking at the prophase. The nucleus has broken down. Those microtubules, as we said, have started to, the MTOCs has formed those spindles. So just looking back at what we just looked at, we've got another illustration here of that. So we've got that nucleolus breaking down, the spindles starting to form, and then our nuclear membrane breaking down. From the prophase, we move on to the metaphase. Okay, at this point, our chromosomes are lining up along the equator of the cell, so that being the center of the cell, the equator of the cell. The microtubules continue to grow until they finally attach themselves to the centromeres, which is the central part of the chromosomes. There are 2 attachment sites on the centromere, enabling the microtubule to attach from each pole. So you'll get 1 microtubule attaching there, 1 microtubule attaching there. So 1 attaching here and 1 attaching here. Tension is then applied from the microtubules to ensure the attachment is correct. So this is all occurring in the metaphase. And now we move on to the anaphase, where the centromeres on the chromosomes divide, allowing the pairs of sister chromatids to separate. So our chromatids have now separated apart. Spindle microtubules rapidly pull sister chromatids towards the poles of the cell, so each end of the cell. This ensures that the end product of mitosis will have exactly the same DNA. Alrighty, so we've pulled those chromatids apart and now we enter the telophase. The chromatids are now called chromosomes. So they're chromatids whilst they're a pair of chromosomes, They're chromatids they're chromosomes when they are alone. Chromosomes are pulled into a tight group near the mTOC, and a nuclear membrane forms around them. The chromosomes now uncoil, and a nucleolus is formed. The cell is usually already starting to divide at this point, ready to enter potentially the interphase again. Cytokinesis, this occurs after mitosis, is the final separation resulting in the two individual cells, and it's different in animals and plant cells. So let's have a look. Cytokinesis in animal cells, the plasma membrane is pulled inwards around the equator of the cell, forming what we call the cleavage furrow. A ring of contractile proteins, actin and myosin, inside the cell membrane achieve this furrow. Okay, so these are proteins that can pull tight as such. When the furrow reaches the center of the cell is pinched apart into two daughter cells. Having a look at this in plant cells, vesicles are formed to the equator where they fuse to form tubular structures. So here, tubular structures across the equator of the cell. They form two layers of a membrane across the whole equator, forming plasma membranes of the two daughter cells. So here we've got mitosis occurring. Now we've got this cytokinesis occurring here. We've got these vesicles lining up, and then they form a double membrane along the center of the two daughter cells. Pectins and other substances are brought via vesicles and deposited between the two membranes, forming a middle lamella that will be the two new cell walls. So that will build up down the center. Vesicles then bring along cellulose to the equator and deposit it adjacent to the middle lamella. Each daughter cell builds its own cell wall then along the equator. This is often referred to as what we call the cell plate. How can we tell if a cell is undergoing mitosis? There's a question. We've got this thing that we call the mitotic index, and it's the ratio between the number of cells in mitosis in a tissue and the total number of observed cells. So if you look here, you can see all of these cells, and certain ones of those are in the stage of mitosis. Okay, so it's calculated using the equation. The mitotic index equals the number of cells in mitosis divided by the total number of cells. So if we're looking at this picture here, you'd count up all the cells, and then you'd do the number of those that are undergoing mitosis, where you can clearly see those chromatids, divided by the total number of cells that there are there. Okay, cyclins are another way we can see if the cells are in mitosis. Okay, so cyclins are proteins involved in regulating the cell cycle. They ensure tasks are performed at the correct time and that the cell only moves on to the next stage of mitosis when it is appropriate to do so. So it's these proteins, cyclins, that control this. They were discovered by Tim Hunt back in the 70s and 80s accidentally. Hunt was researching the control of protein synthesis in sea urchins when he noticed an unusual pattern of protein activity. Further research then led him to realize that proteins were regulating the cell cycle, and then he went on and he named them cyclins. Upon accepting his Nobel Peace Prize, Hunt spoke extensively about serendipity, a happy and unexpected discovery made by accident. Many scientific discoveries have been made this way. Okay, so now we know how cells divide and the stages of mitosis. Let's link this to tumor formation and then look at this through the eyes of cancer. So tumours are abnormal groups of cells that develop at any stage of life in any part of the body. If these abnormal cells adhere to each other and do not invade other tissues or move to other parts of the body, then they're classed as benign. They're not dangerous, they're benign. However, if these abnormal cells become attached and spread elsewhere in the body, then they are classified as malignant. The development of a secondary malignant growth away from the site of the original growth is known as metastasis. Diseases due to malignant tumours are commonly known as cancers. Cancer has many diverse causes. Chemicals and agents that cause cancers are known as carcinogens. Carcinomas are malignant tumours. Things that are mutagens are carcinogenic. Some viruses are carcinogenic. X-rays can be carcinogenic. UV rays are carcinogenic. That should be UV rays, sorry. Many chemicals are carcinogenic. Mutagens cause gene mutation. Mutations then cause cancers. Certain chemicals can also increase the risks of metastasis. Mutations are random changes in base sequences of genes. Not all gene mutations will lead to cancer. The genes that lead to cancer are mutated are called oncogenes. Okay, so remember onco being cancer. Oncogenes regulate the cell cycle. If mutated, the cell cycle gets out of control and tumours are formed. There is a correlation between smoking and the incidence of cancer. Okay, just know that word correlation is the relationship between two variables. So that's one of your key words there, correlation. A positive correlation, as one variable increases, the other variable also increases. And a negative correlation is where one variable increases, the other variable decreases. So do you think that smoking and cancer have a positive correlation or a negative correlation? So let's think about it. The more that a person smokes, the increased risk there is of the incidence of smoking. So it's a positive correlation. Thank you for watching the video on cell division, and I will see you again soon in topic 2.